Good morning, my name is Nicolai van Oers, and I would like to tell you about our recent findings published in the Journal of Clinical Investigation pertaining to mutations in FOXN1, which are classically known to cause effects on the thymus. So to begin with, I'd like to show you a picture of a normal human thymus. This is thymus obtained from a child undergoing cardiac thoracic surgery, where usually a portion of the thymus can be removed. And if we look at this large tissue and take a slice through it and stain with two dyes, H and E, it gives us a view of the thymus as T cells develop in it. We have this very dark purple region, which is a cortical region, and this is where the early immature thymocytes begin rearranging the T cell receptor, with the T cell receptor binding to peptide HLA molecules. And this will determine the specificity of the T cells that develop and mature and escape into the periphery. Upon selection in this cortical region, what are referred to as double positive thymocytes migrate into a medullary region where there are additional purging mechanisms to remove T cells that are potentially autoreactive. Now, this process of T cell selection involves, on the right panel, a population of cells called thymic epithelial cells. These are the cells that present peptide HLA molecules to the T cells, which are the small round balls that permeate and percolate through this developing tissue. The functions of the thymus can actually be assessed by an assay that measures the number of T cell receptor excision circles as a measure of T cell output. Newborn screening includes a test for T cell output for almost every child born in the United States. And this measures the number of these T cell receptor excision circles per spotted blood sample. Low levels of TREC are suggestive of diminished or absent T cells, and this, of course, can lead to an increased susceptibility to infections. For a child born with low TRECs, usually there's a repeat screening to confirm the presence of low TRECs, and in most children, the low TREC numbers will actually stabilize and increase over time. But for those infants where the low TREC numbers are maintained, one usually requires a follow-up to look for potential genetic causes that lead to an immunodeficiency. So we had a situation where two infants presented with low TRECs, and they were subsequently worked up to have a T-negative low phenotype with the presence of B cells and NK cells. And by genetic sequencing, we determined that both infants had distinct compound heterozygous mutations in FOXN1. Now, they had absent to low TRECs. This was suggestive of a thymic hypoplasia, and this connected well with the role of FOXN1, which is the key master regulator of thymic epithelial cell development. But what was unusual in these children was the presence of normal hair extrusion and nail beds, because classically, people have already identified autosomal recessive mutations in FOXN1, and this leads to a Nude SCID phenotype. Nude in the sense that there is no extrusion of the hair follicles through the epidermis, and there's a dystrophy of the nail bed as it expands. These children are born with the absence of a thymus, and this leads to no TRECs and a very severe combined immunodeficiency scenario. So we had a situation where we had compound heterozygous mutations in FOXN1, and the question we wanted to address is whether this was causal to the thymic hypoplasia that we presumed occurred in these children. So this is a structure of FOXN1, which shows in yellow a DNA binding domain, and in blue a transactivation domain. In red are the three mutations that have been identified as autosomal recessive mutations, which lead to a dysfunctional FOXN1, primarily because FOXN1 is produced as a truncated protein or not at all, and there is absolutely no DNA binding activity, and this leads to the Nude phenotype. However, we had a situation where we had a child, in one case, born with one allele with a mutation at a position called 933. This led to a frame shift in the DNA binding domain, and a second mutation on the other allele at a position called 1089, which is just at the end of the DNA binding domain, and this led to a loss of 5 amino acids, but a continuation of an infant protein. And we had a second child who was born with two additional separate mutations on one allele a single nucleotide mutation which led to a change in an amino acid from proline to serine. And again, just before the transactivation domain, a second mutation that led to a frame shift and a truncation of the transactivation domain. So we generated mice that actually matched some of these clinical features in the patients. So this is an example of a wild-type mouse. We see normal whiskers, nice fur, and we generated mice that had autosomal recessive mutations on one of the alleles that was identified in patient 1. And this, of course, leads to a Nude phenotype. But more importantly, on the far right, we generated the mouse that had the compound heterozygous mutations that matched patient 1. And here you can see nice fur or hair, whiskers, there's nail bed. So the question was, we have this normal pattern of hair development. What happens to the thymus? Well, in this, we can obviously see a normal thymus from a mouse is a very large structure relative to this extremely hypoplastic tissue we saw from the mice that genocopied patient 1. If we take these two tissues and compare the developmental processes, in a normal thymus, one can see the double negative cells which lack CD4 and CD8, and these develop into a CD4, CD8 double positive population. This occurs in the cortex, and these cells are selected to recognize self-peptides HLA, which drives them into the CD4 or CD8 single positive populations. In the mice that matched patient 1, we see almost no T cell development. Everything is blocked at the double negative stage. And this is further reflected when one looks then at the periphery. In a normal mouse, one can stain for populations of B cells in the lymph node, marked by B220, and CD3 positive T cells, marked by CD3. In those mice that genocopied patient 1, we see B cells, but absolutely no T cells. We also looked at NK cells, and these mice had a T negative B plus NK plus phenotype, much like patient 1. So what are the clinical conditions resulting in low TREX at birth? One of the primary ones is premature birth, followed by stress load, either in utero or at delivery. In addition, many patients have been identified with gestational diabetes who can also present with low TREX at birth. Genetically, there are a population of individuals who are born with a deletion syndrome called 22q11.2 deletion syndrome. This can also lead to a thymus failure. And finally are the autosomal recessive mutations in FOXN1 that have been identified. These are very rare and lead to the Nude SCID phenotype. But in today, what I've shown you is that there are patients that have compound heterozygous mutations in FOXN1, also rare, but this leads to an atypical SCID where there is no Nude phenotype. And I think the important message of this paper, based on our analysis of many other mutations in FOXN1, is the clinical impact really depends on what mutations exist in FOXN1 and whether they impact either the DNA binding or transactivation domains of this transcription factor. Thank you for your attention and enjoy reading the manuscript.